And dear 12th class students, and I'm your botany sir, and today we are going to discuss the search for genetic material. And previously, it was just thought that the DNA, RNA, they act as the genetic materials, but there was no evidential proof. And then many experiments were carried out to confirm that it is the DNA actually that acts as the genetic material. So, it was the experiment by Frederick Griffith which proved that, which gave the evidence that DNA acts as the genetic material. And now, the experiment he carried out, we are going to take up that. Even though the discovery of nuclein by Michel and the position for principles of inheritance by the Mendel were almost the same at the same time, but that the DNA acts as a genetic material, it took long to be discovered and proven. By 1926, the quest to determine the mechanism for genetic inheritance had reached the molecular level. That means there was advent in the technology or a technological revolution in terms of the equipments and tools that were available to search for the search at the molecular level. And previous discoveries by Gregor Mendel, Walter Sutton, Thomas Hunt Morgan, and numerous other scientists had narrowed the search to the chromosomes located in the nucleus of most cells, that they were of the opinion that it is the chromosomes that carries the genetic material, or the chromosomes act as the hereditary material and carries the characters, carries the duty of transmission of the characters. But the question of what molecule actually was the genetic material had not been satisfactorily answered yet. And now it was the experiment that was carried by Frederick Griffith, and that is known as the transformation or transforming principle. It was in 1928, Frederick Griffith, in a series of experiments with Streptococcus pneumoniae, bacteria responsible for pneumonia, witnessed a miraculous transformation in the bacteria. During the course of his experiments, a living organism, bacteria, had changed in physical form. When Streptococcus pneumonia, that is pneumococcus bacteria, were grown on a culture plate, some produced smooth, shiny colonies represented by the capital S, while others produced rough colonies represented by capital R. This is because the S-strand bacteria have mucus polysaccharide coat, whereas R-strand bacteria does not have this polysaccharide coat. That is why S-strand are smooth, whereas R-strand are rough. Mice infected with the S-strand that was virulent die from pneumonia infection, but mice infected with the R-strand do not develop pneumonia. This is here. S-strand, when injected into the mice, mice die, and R-strand, when injected into the mice live. And Griffith was able to kill bacteria by heating them. He observed that the heat-killed S-strand bacteria injected into the mice did not kill them. When injected a mixture of heat-killed S-strand and live or bacteria, the mice died. Moreover, he recovered living S bacteria from the dead mice, and then he concluded that the R-strand bacteria had somehow been transformed by the heat-killed S-strand bacteria. Some transforming principle transferred from the heat-killed S-strand had enabled the R-strand to synthesize a smooth polysaccharide coat and become virulent. This must be due to the transfer of genetic material. This is the guess that Griffith made. However, the biochemical nature of the genetic material was not defined from his experiments. Here you can see, and this is the virulent Diplococcus with S third type and non-virulent or second type. S is capable of producing infection in the mice, whereas the R does not produce infection. When injected the S into S type this bacterium into the mice died, and when R type was injected into the mice, they survived. They were able to survive. And when heat killed virulent S bacteria were again injected into the mice, they survived. But when together heat killed virulent and the living non-virulent were injected into the mice, some of the mice died because of the virulence that got transformed from S type into the R type of the bacteria. And now this biochemical transformation or the biochemical characterization of the transforming principle. Prior to the work of Oswald Avery, Colin MacLeod, and Maclean McCarthy in 1933 to 1944, the genetic material was thought to be a protein. They worked to determine the biochemical nature of transforming principle in Griffith's experiment. They purified biochemicals, that is proteins, DNA, RNA, all types of the biochemicals which were recovered from the bacterium, and they got those purified from the heat killed S cells to see which one could transform live R cells into the S cells. And they discovered that it was DNA alone from S bacteria that caused R bacteria to become transformed or become virulent. They also discovered the protein digesting enzyme proteases and RNA digesting enzyme RNases did not affect transformation. So the transforming substance was not a protein or RNA. Digestion with DNase did inhibit transformation, suggesting that DNA caused the transformation, and they concluded that it is the DNA that acts as the hereditary material. But not all biologists were convinced, and there were many more experiments that were carried out in the evidence that actually DNA is in the man genetic or hereditary material, and that we will discuss in the coming classes, inshallah. Thanks. Have a nice day.